This summer, imagine a world where there are no genetic diseases. A world free of suffering from diseases such as Huntington's, cystic fibrosis, sickle cell anemia, scleroderma pigmentosum, and many more illnesses. Imagine the possibilities of ending genetic disease as they are being created within the embryo. We will see the amazing scientific advancement known as CRISPR. We will discuss what CRISPR is and what it is used for. In theaters near you. Probably never.
What are you doing, Salma?
Yes, dear?
She's explaining CRISPR. But what is it again?
CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a set of DNA sequences found in the genome of prokaryotic organisms, such as bacteria and archaea. These sequences are derived from DNA fragments of bacteriophages that have previously infected the prokaryote. Thus, they seem to have been artificially vaccinated, as that is their immune system.
Well, not so fast. CRISPR was initially made as a defensive tactic to defend bacteria against their viral intruders, the bacteriophages. Bacteriophages are viral particles that infect the bacteria. When infected, the bacteria are figuratively vaccinated.
It's time to get crispy.
Did you just really say crispy?
Maybe.
I just don't know anymore. Why?
Anyways, I remember now. CRISPR consists of two components, the Cas9 protein that can cut DNA and the guide RNA that can recognize the sequence of DNA to be edited. To use it, scientists look for the problematic DNA sequence which must be edited because it is causing a health problem. Then, scientists will create a specific guide RNA which recognizes that sequence in the problem region of the DNA. The guide RNA is then attached to the Cas9 component. This Cas9 and guide RNA complex is then introduced to the target cells. Once inside the target cells, the complex locates the specific problem DNA sequence and cuts it. From here, scientists can edit the existing genome by adding new sequences, modifying the sequences, or deleting the sequence.
Dude, just say that CRISPR is a fancy cut and paste tool, but in real life.
So there's a fake life?
Yeah, I know, David. Anywho, some bacteria are known to contain one or more CRISPR regions while others have none. The CRISPR loci is quite extensive, containing over 100 repetitions and spacers. The primary functions of CRISPR are 1. Acquisition, also known as adaptation, is the process of obtaining DNA fragments from a phage. And 2. Interference, which is the immunization process by which Cas proteins are encoded by Cas genes.
Hey guys, I can say smart stuff too. Just watch. Back in 2012, one of the key findings that contributed to the use of CRISPR systems was known as genome editing, which identifies proteins involved in the cleavage of hybrid DNA. The procedure was conducted by a complex of proteins which contained Cas1 and at times a single protein Cas9. Cas9 is unique in the sense that it attaches itself to a DNA strand, and due to two distinct domains in the structure, it is able to cut the DNA on each of its two strands. The Cas9 protein is the basis of CRISPR-Cas9 technology, which, in addition to bacteria, allowed for a range of modifications of genome and mutations in eukaryotic organisms, such as plants, insects, mammals, and fish. The CRISPR complex is made up of the Cas9 protein and a hybrid guide RNA molecule. This RNA molecule is made up of RNA similar to the region that is mutated and a second RNA called tracker RNA. The discovery of the tracker RNA was found alongside CRISPR locus in Streptococcus pyogenes and was shown to be similar to the repeating locus regions, serving as a guide for Cas9 proteins and crRNA towards the target.
To add to what Ricky the interrupter said, there are 3 steps with CRISPR. 1. Integration of a piece of DNA from phage into CRISPR locus, acquisition. 2. Expression of Cas proteins and pre-cRNA, which split small cRNAs. And 3. Interference occurs when DNA is inserted into a bacterium by a phage and reaches the cRNA region, which deteriorates the hybrid form, therefore preventing infection.
Now, if I may take over, Rico, because Ricky also interrupted me.
By all means, Alex, I understand how he just interrupts.
Thank you. The Cas9 protein named DCAS or dead Cas, when fused to repressor and activator protein, can adhere to desired locus without dissociating it, thus controlling the gene expression in animals.
The CRISPR method, however, can introduce multiple mutations in genes. The targeted gene therapy is the culmination of medicinal advancements in CRISPR methodology that have major clinical application and implications in medical treatments. Proteins in the bacteria are endowed with many functions. They allow DNA fragments to associate into the CRISPR loci and also mediate responses to bacteriophages. Upon injection of the bacteriophage DNA into the bacteria, the small RNAs bind and an enzyme dissociates the phage from the RNA. Infection is halted.
All right, my students, this is nice and all. You've explained what CRISPR is, but can you give me an actual example of what it's used for?
Of course. An excellent example of CRISPR's use is its utilization in the dairy industry. Within the dairy industry, there are certain bacteria such as Streptococcus thermophilus, which transforms the lactose in milk into lactic acid, which is used to make yogurt and cheese. On another note, each different type of bacterial strain which is used to make yogurt contributes to a unique texture and taste that the yogurt has. Thus, if a bacterial strain is eradicated due to a bacteriophage infection, this spells out economic disaster for the manufacturer of these products, and thus proving why CRISPR proves to be an essential tool nowadays.
Correct, Ruben. Very interesting indeed, my dear students. I would like to know what are some of the future applications of CRISPR. Would you mind elaborating?
In 2017, the Oregon Health and Science University used CRISPR to remove a defective gene from a heart condition known as hypertrophic cardiomyopathy. Thus, CRISPR's future work will be delving into correcting genetic errors which may cause a myriad of diseases such as HCM.
In addition to that, CRISPR is being utilized to try and eliminate microbes which cause diseases such as HIV. Nothing successful has happened to date, however, this may soon change and thus sporadically efface diseases that were once thought to have no known cure.
To add to that, CRISPR is being investigated to see if it can be used to resurrect extinct species. For example, back in 2017, Harvard geneticist George Church was trying to use CRISPR to resurrect the woolly mammoth. However, it would be a hybrid between the mammoth and Asian elephant.
Another exciting potential use for CRISPR is to enhance current foods. Since CRISPR is a fancy cut and paste tool, it is currently being investigated if it can be used to try and make grown fruits and vegetables bigger and give greater yields.
Lastly, CRISPR is being studied for its possibility to fight pests. Scientists are working on manipulating the genes of pests, such as mosquitoes. As you may well know, mosquitoes are disease-carrying and are able to spread their diseases very effectively. Imagine the possibility of being able to eradicate mosquitoes from certain areas of the world, which are prone to illnesses given off by them. However, this may come at an unforeseen cost, being that mosquitoes make a great part of the food chain.
Well, that is pretty much it for CRISPR. We hope to have enlightened all of you with some marvelous information for what CRISPR is, what it is used for, and possible future work.
Remember, stay crispy!